Hello, this is Tommy. Welcome back to Chatomics. In today's video, I'm going to show you how to make an offset plot in R. And make sure you stick to the end because I'm going to walk you through the code step by step using an R package called ComplexHeatMap. So what is an offset plot? So the offset plot looks like this. Okay, it is used to visualize sets or different sets of genes, for example. We all actually are very familiar with the Venn diagram, right? For example, when you have two sets of genes and you want to know, okay, what's the intersection of those two sets, then use this Venn diagram, two-way Venn diagram. But when you have, for example, 5 sets of genes, the Venn diagram looks like a little bit funky. For example, if you look at here, when you have 5 sets, and it can be a little bit more too complicated to interpret. And offset plot gives you an alternative way to visualize it. So in this example, and as you can see here, this is from a genomic sequencing paper. So you sequence a bunch of samples and then you identify mutations in those samples. For example, you have those commonly mutated genes, RB1, EGFR, TP53, TP10. And in this plot, what is showing here is, for example, if you want to look at the co-mutation of two genes, EGFR and the P10, then you go to here, then you go to the bar and you see, okay, there are 16, for example, samples or patients that have both mutations. And for example, you only have actually 2 patients have this RB mutation, PIK3R1 mutation, TP53 mutation, and P10 mutation. Okay, this just gives you a much better way to actually visualize the results when you have many sets. So in this case, 1, 2, 3, 4, 5 different sets, right? And in fact, actually in my paper, so this is a paper that I published in 2021, and the first figure I showed is an offset plot. So in this case, actually, so each set is 1 sample here, but I have actually 2 tumor samples from the same patients. So patient 26, you have tumor 1 and tumor 2, and then you look at tumor 29 here, you actually 3 different tumors from the same patient. And here, the actually number here showing actually the number of mutations that are shared across different samples. And here is just the total number of mutations in each sample. And as you can see here, most of the mutations, they are actually shared within the same patient across different samples, right? As you can see here, these 3 patient samples actually from the same patient, then they have the most common overlapping mutations. And these 2 from the 37, those 2 from 26, those 2 from patient 31. Okay, so offset plot gives you a very nice way to visualize different sets. So especially when you have a lot of different sets. So I'm going to show you how to code it up in R, and actually you can use many different packages. So the most original one is the offset plot R. I think it's called offset R package. And you can also use the later developer that I think called ggOffset. But I'm going to use this tool called ComplexHeatMap. So this actually, ComplexHeatMap is actually almost exclusively used to make heat map actually in my daily work. But because essentially you can consider this as a heat map body, and those bars, those are just top annotation versus the side annotation. So the tutorial ComplexHeatMap reference, and there's a specific section describing how to use ComplexHeatMap to make offset plot. Okay, so let me actually walk you through the code step by step using gene sets as an example. Okay, we're going to actually use this mseqdbr package, which contains all the gene sets from mseqdb. So if you don't have it, you can install it. And let's load the library, dplyr. And then you can actually query the gene sets using the mseq-dbr function, specify the species. You can also specify mouse, for example, and then the category will be the hallmark gene sets. Okay, so if we do this, and this is actually a tidy data frame, and you take a look. So each row actually is one gene. It will tell you, okay, this is the hallmark set's name, and then this is the gene symbol, untrans ID, ensemble ID, yeah, things like that. Okay. And if you look at the gene set name here, actually there are total, I think, 50 hallmark gene sets. They are all prefixed with hallmark. Okay. So what we are going to do, because now it is actually a tidy data frame, so we need to actually get for each pathway or gene sets, we need a list. So if you go to the tutorial of the set, you will see here the input will be a list. So you have set 1, then you have the values of that set 1 and set 2 and the value of set 2. So in our case, this will be the gene set name, and then those will be the gene name for that set. Okay. So how do you do that? Okay. So we can use the tidyr nest function. So if we run group by the gene set name and the next nest into a list column. So if you look at this data frame, now you have a table with 2 columns. One is the gene set name. So we have actually 50 gene sets. And then we have the data column, which is a list of data tables actually. So we can actually take a look at the first element of the gene sets by using this. So the gene set nest, this one, and use the $sign to access the data column. And because that column is a list, right? You see this is a list of tables, and you can use just the double bracket number 1 to access the first table. Okay, so if you do this, it just show you, okay, this is just gene set 1 and with 210 genes for that gene set. And what we really need is just this column, gene symbol, right? So what we can do, we can use the map function to loop over this column and only pull the gene symbol column from each table from here. Okay. So let's see how it looks like. So if you do this, and now this gene list will be just a list of genes in each gene set, and you can see we have actually 50 here. Okay, so because then there's no name here, okay, because we want to actually add the gene set name as the list name. So what we can do is, because this is the data frame, if you remember, 2 columns, and then we just get the gene set name, and because they all have the hallmark, so I just want to remove them. So use the string marks, the underscore replace the hallmark underscore to empty string. So essentially we'll remove the prefix and then give that list those names. Okay, so if you will look at this, now the list actually has names. Okay, so if you just take a look, it will be the same list, right? But now for each element, you have a name, which is that gene set name. Okay, and just for the demonstration purpose, I'm going to just take actually 5 gene sets from the 50. So I took actually 13 E2F targets, 14 epithelium to mesenchymal transition, estrogen response early, estrogen response late, and the G2M checkpoint. Okay, so I only subset 5 elements from that list. So we do this, gene sub, and then look at the name. So those are the 5 gene sets we're going to visualize. So essentially we want to see, okay, for those different gene sets, how many genes are common between different sets? For example, this estrogen response early versus late, like I bet there are many actually common genes in those two sets, right? Okay, so in order to do that, so we actually load the library, ComplexHeatMap, and there is this function called make combination score mat, and then the input will be just the list, okay, this list, and give it to this function and take a look. So essentially it give you this actually combination matrix with 5 sets, right? Because we have 5 gene sets and 17 different combinations, okay? So now that we have that combination matrix, we can just feed it into the upset function within ComplexHeatMap. Let's take a look at how it looks like. Okay. So essentially this is how it looks like. And then as you can see, for example, E to F, target, and G to M, they have like a lot of actually almost 100, like maybe 75 something overlapping genes from those two sets. And the early versus late, those two estrogen response, and let's see how many. They also have quite a bit actually overlapping, almost 100, right? So this is a very good way to visualize, right? And it also have very small number that are common, like for example among 3 different sets of gene sets. And by default, this is ordered by the number of actually common ones. Okay, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, and then you have the unique actually sets. But we can actually reorder them by, for example, by the number of the size of the sets, for example. So this one will be arranged into the very last or very first. You can do that by using this, the combination order equals to the combination size. This is the help function from ComplexHeatMap. If you do that, it'll tell you how many actually combination for each combination, how many genes for each combination. Okay, so in this case, we are order from small to big. Okay. If you do order, so this is just the size, and if you do order, and then if you do this, and then you will see now it's actually arranged by the size of the sets, okay, from small to big, okay. Of course, you can just reverse it by just actually add a minus sign here, for example, if you do that. And then if you take a look, and now you have the biggest one actually in the front, okay. So it's very flexible. Again, like I was actually guessing just how many genes common between sets or among different sets. So we can actually add a number on the bars. So by using this one, so top annotation, upset top annotation, the matrix, the combination matrix, and then you can specify add numbers equals to true. Okay, and if you do this, now you have the numbers actually on top, so it makes it a little bit easier for you to actually see the exact number of common genes. Okay. I already showed you how to actually change the order of the bars, but you can also change the order of the sets. For example, here I want those two estrogen together, estrogen related response gene sets to go to each other, and also E to F and then G to M together, for example. So I can just use the set order equals to this, so you can specify the order you want to order them. Okay, so E to F, G to M, and then the estrogen response early and late. And then lastly will be the epithelium to mesenchymal transition. Let's take a look. Okay, so now you can clearly see, okay, those two are close to each other because I know there are many common genes for those two sets, right? And then again, similarly for those two sets, estrogen receptor, estrogen response early and late, you have like 95 genes that are common. And this is just tell you the total number of genes in each set. I think it's 200 for all of them, just by coincidence, okay. And Okay, I already show you. Okay, you can actually reverse the order from high to low like this. Okay, and also, and you notice that these row names here, because I think the font size is too big, and you get cut off here. So you can actually change the row name size by using this row name _GP. Then you can specify the font size to 8. Okay. So this is actually the parameter that you can specify within the heatmap function, for example. So if you look at the help page, and you see those are the available arguments that you can specify. Then you also have ... So essentially those are the other arguments that pass to heatmap. So if you click this, and you will see the row name. If you say row name, GP, for example. So exactly, so you can specify the size of the row name. So I'm using font size 8 here. So let's see how it looks like. And now the font size becomes smaller, so you don't get it cut off, right? Because this is a bit longer gene set name here. Okay, so this just show you how actually flexible ComplexHeatMap is to make upset plot. So you can actually add many other stuffs here, because essentially you can think about this as a heat map, and then you can add many other different actually annotations. So if you go to the tutorial, and you will see, if you scroll down, it's a very long actually tutorial. You can change the different colors, right? You can actually have multiple upset plot actually combined together, either vertically or horizontally, and you can change the bar to whatever shape you want to have, even like this. And you can also add those like heat map type of annotations, like this bar annotations on top or on the side. And you can also add like other type of actual plots that is supported in ComplexHeatMap. Okay, so it's a really powerful package, ComplexHeatMap. It's not only used for making heat maps, but it can be also used to visualize sets. So I highly recommend you learn how to use ComplexHeatMap. And there's always a trade-off between usability versus like flexibility. So if you really want to quickly make a upset plot, maybe you can use other package called upsetR or ggset. So you can easily create a plot very fast, but then it lacks flexibility, right? If you want to have everything in control, then I will use ComplexHeatMap. So there's always a trade-off. So if it allows you to have a lot of extra flexibility, then you need to write a lot of custom code. But if you can just easily make some plots, but it's not really easily configurable. Okay, so that's it for today. I hope you learned something, and make sure you subscribe. I will have the scripts also in the description of this video, so you can actually follow the tutorial. And that's it for today, and happy learning. I will see you next time.